All right, good morning or afternoon, depending on what class you are in right now. We're going to go ahead and start our next unit. And from here on out, we're going to be covering the eukaryotes. So everything we talk about at this point are going to be domain eukarya. And so we're going to start with the protists, and then we're going to cover fungi. And so these two together will be a unit, and we'll have tests and activities over the protists and the fungi. Then we're going to move to plants, and we'll have a whole unit on plants. And then we're going to move to animals, and we'll kind of end the year with animals and like body systems. And so with the protists, these are going to be our first eukaryotes. And so one of the things I want to kind of briefly mention, we didn't really talk about this too much with evolution, but we've spent a couple of weeks at least talking about prokaryotes, and we know that prokaryotes are unicellular. They don't have nuclei, they don't have membrane-bound organelles. They include the bacteria and the archaea. Of course, we know that viruses are not living, but we kind of included them in that microbiology unit. And as we continue, we're still going to be in an area where we could kind of consider this microbiology, but we are moving to these eukaryotes. Well, remember that all things in biology can be explained through evolution. And so one of the main widely accepted theories that proposes how did we get from the unicellular, less complex prokaryotes to the more complex eukaryotes. And the most widely accepted idea is known as the endosymbiotic theory. And so if we break this term down, endo means inside of, or inside, and then symbiotic refers to those close relationships. And so essentially what the endosymbiotic theory suggests is that prokaryotes came first. And so before any eukaryotes existed and there were only prokaryotes on the planet, some of those early prokaryotes, those single-celled prokaryotes, would engulf or take in, by phagocytosis, they would kind of surround other prokaryotes and engulf them. And then instead of bringing them in and digesting them, they would bring them in and kind of live in a symbiotic relationship. So the smaller prokaryote would provide a benefit for the larger prokaryote, and the larger prokaryote would provide a benefit. And then after generation and generation and generation, kind of like what we see in our pictures, is as the cells began to replicate, they would also replicate these internal little structures. And that's how we get some of our modern day organelles. Now the main evidence is in two main organelles, and that's the mitochondria and the chloroplast. And so it's believed that they were tiny prokaryotes. The mitochondria were prokaryotes that could do aerobic metabolism, and the chloroplasts were tiny prokaryotes that could perform photosynthesis. And so as they became engulfed, and over time, because they weren't broken down, they kind of became little cells inside of cells. Now today, if you were to take the mitochondria and chloroplast out of a eukaryote, they could not survive independently, but it's believed that at one point they were small prokaryotic cells that could live on their own. And so the main evidence for that is we see these modern relationships, these mutualistic relationships of organisms living inside of other organisms. But in terms of the mitochondria and chloroplast, the reason they are the main organelles that are believed to have come from early prokaryotes is because they're about the same size as a prokaryotic cell. They have these inner membrane structures. They also have a double membrane, which suggests a membrane being engulfed by another membrane. They have their own enzymes. They can replicate themselves. Now they have their own genome, and what's important about the genome is just like bacteria have like circular pieces of DNA, right now in our cells, mitochondria and chloroplast have their own circular DNA. And then ribosomes, of course, for protein synthesis. So they're literally little cells inside of our cells, and that's what supports this concept of prokaryotes engulfing other prokaryotes in the endosymbiotic theory. And so the first eukaryotes, and the simplest of the eukaryotes, would be your kingdom Protista. It would be your protist. And for a very long time, they were put into kingdom Protista. Now, after further, study and looking at DNA analysis, honestly, I think taxonomists and phylogenists don't really know exactly what to do with protists. They are no longer considered a true kingdom, but in a lot of resources, you will still see kingdom Protista. But essentially, how do we define protist, or how do we define the kingdom Protista? They're the eukaryotes. This is the one thing that they all have in common, but they're the misfit eukaryotes. They're the eukaryotes that don't fit into the other eukaryotic kingdom. So they're eukaryotes, but we can't put them in the fungi group. We can't quite put them in the plant group, or we can't quite put them in the animal group. So the one and only characteristics that all protists share, is that they are eukaryotic cells, which means they do have either a nucleus or multiple nuclei, and they do have those complex membrane-bound organelles. So that is definitely what sets them apart. A lot of times they will get confused with prokaryotes because many of them are unicellular, but they do have the components that make them a eukaryote. So this is the one defining characteristic that sets the protist apart from those bacteria. Now, pretty much everything else, there's going to be exceptions to the rule. As we go through characteristics, what you're going to find is that there's diversity, and there's more diversity. When it comes to that group, it's kind of hard to kind of sum them up like we could a plant or an animal or a fungus, and that's why they kind of get put in this misfit kingdom. So structurally, they're very diverse. They can vary in structure. They might have a cell wall. If they do, that cell wall could be like a plant, or that cell wall could be like a fungus. They could have these other external structures like a shell or a test, and they could be made up of different components. Some are glass-like. Most of them are going to be unicellular, but they could live in colonies, or they could be multicellular. Some of your algae groups are enormous, like certain kind of kelps, and so while most of them you have to have a microscope to see, there are some that are taller than you. Most of them use aerobic metabolism because most of them do have mitochondria, so they're able to use oxygen and perform cellular respiration. And then a lot of times they have other organelles that you may not even find because they live in different places, and they may be aquatic, and they may need special pumps or things like that. So one of the things, the way we're going to cover this unit is this is going to be a very short set of notes. It's kind of an overview. But over the next several days, you have different activities that are going to expose you to many different types of protists and give you a little bit more information. And it's going to help you to see the diversity within this group. Here we have the word diverse again. So when we look at their nutrition is diverse. So we have some that are going to be photoautotrophs. They could perform photosynthesis. We have some that are heterotrophs, which means they have to eat, or they are decomposers. And then there's some that kind of do a combo where they can do both photosynthesis and consume. And so based on nutrition, and this is what you're going to see a lot over the next several days, is that we can put them in kind of three main categories based on how do they get their food. They could be animal-like, and we commonly call those protozoa or protozoans. And these are the ones that are your heterotrophic consumers. Down here, this would be this holozoic. So these are the ones that eat, like an animal would. They could be plant-like protists, and these we commonly call algae. And sometimes the algae are kind of combined with plants. They're very much plant-like. They have chlorophyll. They have the cell walls of cellulose, but they're grouped together because they can perform photosynthesis. And then you have your fungus-like protists. So these would be heterotrophic, but they're what we call saprobes, or they're absorbers. So they don't physically consume, but they literally absorb their nutrition. And so as you go through your activities, that's one of the ways that you're going to see these protists divided up. All right, when it comes to motility, once again, we're going to have diversity. Most of them are motile, which means they can freely move around, but there are some that don't move around at all. For those that are motile, they may have flagella or cilia. And then there are others that have these structures called pseudopods, which means false or fake feet, like this amoeba here. And you're going to see more about those over the next coming days. When it comes to their reproduction, once again, we have diversity or variation. These are eukaryotes, which means when they do cell division, it'll probably include mitosis. For some of them, that may be the only way. They could be completely asexual. Remember, mitosis gives you the same. Now this binary fission here would not be the same binary fission that we saw with prokaryotes. Remember, prokaryotes didn't have a nucleus, but our protists do. So if they were to kind of copy and divide in two, it would be by the asexual process of mitosis. They could bud, where they literally kind of grow a new one and then it pinches off, or they could create spores that could develop into a new organism. Once again, very diverse. There are some sexual methods of reproduction among the protists. They can produce gametes by meiosis, and those gametes can fuse to form a zygote. Some, like your paramecium, can conjugate. And this is going to be similar to the conjugation that we saw in bacteria, but this time they're not exchanging plasmids. They're exchanging whole nuclei. That's something that would be different from a prokaryote, because they have the plasmids and we don't. And then eukaryotes have the nuclei and they don't. And then some can actually form these cysts, these kind of resistant cells, and they can withstand very harsh conditions and wait for better reproduction, environments. And so here's just an example. Here's an example of binary fission. Here's a couple of examples, one asexual, one sexual. And there are going to be some protists that they may use a combination depending on environment and, nutrition and things like that. If times are good, they may use sexual reproduction. If times are not as good, they may do asexual reproduction. So some of your protists could flip between asexual and sexual. Here's an example of conjugation. This is common among the paramecium. And so there's a wide variety of reproductive methods. When it comes to habitats, typically they're going to be either in water, they're going to be aquatic, or they're going to be in areas where there's moisture, like damp soils. Because they're unicellular mostly, they're going to have to be in somewhere where there is at least some amount of water. Otherwise, they would dry out and desiccate. So they could be bottom dwellers, they could be free swimming kind of in freshwater or marine. They're very important components of plankton. And plankton are kind of a big deal when you look at food webs in aquatic and ecosystems. And so they could be these tiny floaters. Now there are two types of plankton, and you may come across these terms later on, but you have phytoplankton. These are producers, these are autotrophs. And then you have zooplankton, which are animal-like. These would be consumers. But together, all of these are tiny floaters. They're microscopic, and they just kind of drift along in the body of water. And then we're going to see as we go through the unit that many of your protists could be harmful. They could cause disease. They could be symbionts, and most of them would be parasitic, where they steal nutrition from a host. And so if they're living inside of a host, a lot of times they're living within the cells and tissues, which have fluids. And so no matter what, you're not typically going to find your protist in dry environments. And then the last little slide, I don't expect you guys to know all of this specifically, but if you were to look at modern classification, and honestly it could have changed since I made this PowerPoint, but if you look at kind of your more modern classification of protists, they're put into these different phylogenetic groups. And as we go through the next several days, you're going to be exposed to many different examples, things like diatoms, or amoebas, or euglena, or paramecium, which are a ciliate. And so I don't expect you to know all of these kind of very specific terms, but I do want you to be exposed to the diversity that you will find in Kingdom Protista. And so that's why we're going to do a lot of different activities that will help to kind of expose you to all of the different organisms that you could find in this classification. And that's really all you need to know for now. Like I said, you're going to have several different activities, and hopefully you'll get to learn a lot about different protists and get to be more familiar with them. In my opinion, they're probably the least well known of all of your kingdoms, because we don't really see them a lot in our day-to-day life. Even though they may be present, we don't see them like we see plants, or we don't see them like we see fungi growing in our yard. And so I think a lot of times students, they don't have a lot of background knowledge. So we are going to spend a lot of time learning about the variety of protists that exist. I hope you guys are having a great day, and I hope to see you soon. If you have any questions or concerns, please feel free to reach out to me. Y'all have a good one.